Hello, we are Team 7, and today we'll be presenting our project on optimizing bone scaffold regeneration by mimicking bone structure utilizing host cells and 3D manufacturing with my teammates Colton Chung, Amy Nunn, Brittany Pham, and myself, Ryan Sanger.
So annually, around 500,000 patients require some sort of bone repair or restoration in the United States. This being said, around 1.5 million individuals suffer due to bone diseases, such as osteoporosis, and one of the biggest issues from bone disease are fractures. This issue is expected to affect 1 out of 2 Americans over the age of 50, so it leads to an annual cost of over $2.5 billion, with a double amount near the year 2020 due to the baby boomer generation coming to a median age. When the patient has a fracture that is unable to heal itself, they will go through a procedure of having a bone graft, which requires a section of the patient's bone to be removed and then have a 3D bone scaffold put in. There is a pressing issue with the current bone scaffolds in the market, as they have a strict geometrical formation, affecting the quality of the scaffold itself. So, as a theoretical solution, it is important to have a bone scaffold that mimics the pore structure of the human bone, allowing for a smoother fusion from scaffold to bone. It is important to fulfill the needs of a rheumatologist, for example, as they are part of the audience that would use the product to treat the patients in need. However, the target audience would include individuals coming to an older age, solely having the risks and symptoms of osteoporosis, and those who have undergone a severe trauma and are in need of a bone repair.
As said in the last slide, there are an estimated 1.5 million patients each year with degenerative bone conditions, needing implantation that will encourage the regrowth of the degraded bone, such as osteoporosis. Osteoporosis is a common bone disorder characterized by the progressive loss of bone. Bone tissue naturally degrades and renews itself. However, with osteoporosis, the bone degrades faster than it can regenerate. With current scaffolds' osteoconductive properties, it enhances the implementation of the device and success rate to combat these conditions.
So, bone scaffolds, as stated earlier, are 3D biomaterials which allow for a more promising surface for proper regeneration of tissues. It is a matrix that mimics and allows for the attachment of osteoinducible cells to any necessary surfaces. The fabrication material generally includes those that are inorganic as well as organic. For the scaffold itself, the structural aspect is crucial, as there should be interconnected pores and a higher porosity. With the scaffold's limitation of such a rigid geometrical formation, the pore size should be big enough to allow the cells to move and migrate properly and allow for the binding of the cells to the scaffold. However, there is no even distribution of the cells as they are added post-treatment of the scaffold being placed in. So ideally, a scaffold should be biodegradable as well as obtain the necessary mechanical properties. Many scaffolds used are made similar to hydrogels, which are insoluble in water and can mimic living tissues. However, they are fairly weak in regards to mechanical properties, making them difficult to handle and continue to be unsustainable over time. So, to help combat this issue, composites of polymers and ceramics are used, and natural materials such as polysaccharides will help with any cell adhesions.
During our research, we discovered certain criteria must be met. The engineering criteria for a bone scaffold must be biocompatible, which creates a limited immune response. It must be biodegradable to promote cell attachment, proliferation, and differentiation. And the structure of the scaffold must mimic bone structure, as well as the mechanical properties. These properties are Young's modulus between 16 and 20 gigapascal, compressive strength of 131 to 224 megapascal, tensile strength of 79 to 151 megapascal, shear stress of 51.6 megapascal, and an overall porosity of the scaffold of 5 to 10%. The scaffolds need to be sterilized prior to the implantation without losing any osteoconductive and osteoinductive properties.
As most bone scaffolds have rigid structures which do not withstand the varying shear and torsional forces applied to scaffold post-implantation, such as lattices or hexagonal shapes. In order to improve upon current bone scaffolds, our design is meant to be an easy transition into the patient's biological system by mimicking the pore structure of the bone as much as possible and utilizing the host cells in the scaffold. By mimicking the structure of the bone and utilizing host cells, will optimize biofunctionality as the cells would be compatible with a similar shape and recognize their own kind. This allows the cells to proliferate and minimizes immune response. As I stated in the last slide, some scaffolds may have square or hexagonal shaped pores, which we look at an SEM of human bone tissue as more rounded structures and is less uniform. Our group made the scaffold on the bottom as similar as possible with different pore sizes using N-topology. N-topology is a CAD program widely used in creating scaffolds due to its free range when it comes to pore structure. The scaffold can be then customized for each patient using this CAD program. Our scaffold will be then 3D printed using a composite of HA and TCP, FN coating, and the cell serum simultaneously. For printing them simultaneously with 3 different nozzles, it avoids defects and flaws such as non-uniform distribution of cells. HA and TCP composite would be extruded from one nozzle, FN the second nozzle, and the cell serum the third nozzle. As the scaffold material prints, the cell serum and FN coating will be integrated into the pores and walls of the scaffold. HA and TCP are raw ceramics that show high similarity to bone minerals and mechanical properties of bone, which makes them biocompatible, biodegradable, and osteoconductive. Since HA does not have the same mechanical properties as bone, by adding TCP, it brings the mechanical properties of the scaffold closer to bone. HA and TCP degrade relatively slowly, allowing time for regeneration before it degrades. After implantation in the host, they stimulate osteoid formation onto its surface, which is the first step in bone remodeling. Fibronectin is a very common, non-specific, and cheap material used in biomaterials. FN promotes cell adhesion to all cells via a cell binding domain. Mesenchymal cells are the stem cells of the bone, which give rise to osteoblasts, osteocytes, and osteoclasts. The cell serum will also contain nutrients needed by the cells, which will contain carbohydrates, proteins, fats, vitamins, minerals, and water. We'll also promote angiogenesis by having vascular endothelial growth factor, or VEGF, in the cell serum, which is a signaling protein that promotes the growth of new blood vessels. To acquire mesenchymal stem cells, cells would be taken from the bone marrow of the host, and the stem cells would be separated via RCB lysis. RCB lysis shows a greater amount of human mesenchymal stem cells that can be isolated from 10 times less bone marrow volume of 6 milliliters than density gradient centrifugation, which needs 20 to 60 milliliters of bone marrow. Density gradient centrifugation is also difficult to standardize and is slower than RCB lysis. We would then culture and see the scaffold via bioprinting at about 50,000 cells per milliliter, depending on different patients' needs.
During the analysis, we discovered that the value of the solution would be that the patients will not need another graft to replace the implant because of the increased functionality of the design. Increased induction of the material due to FN coating, HA, and TCP, and patient cell infused 3D printing helps host cells adhere to the scaffold. Biofunctionality is increased due to the use of HA, TCP, and the host cells. The increased function of the dynamic nozzle will reduce the need for changing the nozzle, which increases the time it takes to create the final product. Cost of the solution, or estimated cost for the quality of biological 3D printer, is roughly $150,000 based on the present day technologies. The cost of creating the bioink is roughly $500, which involves culturing the patient's cells, then placing them in an incubator, which slowly mixes the cells into an ink. Bioceramics, HA, and TCP can be purchased for $4.72 per gram, starting at $118 for 25 grams of the powder region. Current limitations to this are current CAD software is limited to the porosity it can produce, such as programs like N-topography. While the imaging or design of a bone scaffold appears to be similar, it lacks the capability to manufacture these porosities. Presenting bioprinting methods lack nozzle ability to alternate between nozzle size, which can make it more difficult to get accurate porosity for the scaffold.
Next, we'll be moving on to the test we will conduct on our device. So to evaluate the mechanical properties of the scaffold, an Instron A872 will be used for a compressive and quasi-static test. There will be a total of 6 tests, 3 compressive and 3 static, quasi-static tests. The specimen will be placed between 2 platings of a 1 kilonewton load cell. The compressive test will run at a strain rate of 1 millimeter a minute until failure. This will give the modulus of elasticity, yield strength, and stiffness of the device. This will also allow us to observe the scaffold's failure to ensure it is not catastrophic in vivo. This quasi-static test will also run at 1 millimeter a minute and will cyclically load the specimen around 100 newtons to test fatigue. As mentioned earlier, cells must adhere and distribute within our bone scaffold. To test this, the device will be printed with the cell serum and be placed in a suspension of Osteo1 cells for 24 and 48 hours. After seeing the cells, the implant will be observed with a scanning electron microscope to analyze surface properties. The SAM will allow us to observe the cell absorption and proliferation on the surface of the pores. We will also look at the cells in different regions of the device to inspect whether they are evenly distributed. Next, the scaffold will be printed with the cell nozzles and observed with the SEM. Our team will then compare the adhesion and distribution of the printed cells to the seeded Osteo1 cells. After evaluating the cell response on the surface, our team will understand if our process is sufficient or better than seeding cells as a post-treatment. To measure growth factor release of our device, we will use an Accurus smart reader to measure absorption values. The standard curve will be created with known concentrations of VEGF. The growth factors will be diluted with DMEM. First, the device will be loaded with a serum only containing the growth factors. It will then be placed in a DMEM media for 12, 24, 36, and 48 hours. The media will then be placed in the wells of the plate along with a standard curve to calculate the concentration using the absorption. Next, we'll be moving on to our animal models. So pigs will be used for both our large and small animal models. The Danish Lanzera is a common domestic pig breed that will be served as our large animal model. The Guten King mini pig is a breed commonly used in laboratories for medical research. It will be our small animal model. Pigs are great candidates for testing because they have similar genetic makeup, physiology, and size to humans. For example, their femurs have similar mechanical properties and cross-sectional area to human bones. This makes them ideal to stimulate a mechanical environment for a device. In the pharmaceutical field, pigs are also often used in preclinical trials to test toxicology. They have similar metabolic pathways to humans, making them suitable to test an immune response, nutrient release, and bone cell induction of the scaffold. Here are our cited sources, and thank you everyone today for your time, and we look forward to having your questions in class tomorrow.